Okay, we've just described how we name our different kinds of organisms and group them into different named groups. This hierarchical pattern of kingdom, phylum, class, order, family, and genus and species. That's very convenient for us to make reference to our different groups and name them and identify them, but there are some problems with that, so we're going to talk about trouble with taxonomy. And the first thing we're going to identify is how that misrepresents the phylogeny, that hierarchical naming system that we have. So let's say we have over here the different kingdoms. We have a kingdom, Protista maybe, then fungi, then Animalia and Plantae. So you have all these different kingdoms, and they're on their own paths here, so these are different kingdoms. And from the animal kingdom here, you're going to have a number of different phyla. There's going to be sponges, cnidarians, flatworms, roundworms, arthropods, mollusks, annelids, echinoderms, and somewhere in there we'll get our chordate phylum. So here's our phylum here. And we know from the phylum, next we'll have classes. So there are different agnathan or jawless fish groups. There's the chondrichthyon, there's the ray fin fishes, sarcopterygians, reptiles, amphibians, reptiles, birds, mammals, all that in here. Let's pick on the amphibians. This is a class right here, right? This is a class. Amphibia coming out here. So we've had Animalia, Chordata would be the next one, then Amphibia over here. And then from this class, we have the different orders that come out from that. And the class Amphibia, we have some extinct classes, but in our modern classes, we just have 3 here. From these classes, we have, we have 3 orders from the Amphibia. The frogs, the salamanders, and the caecilians. I like to work with salamanders, so we'll pick on the salamanders. From the salamander group, we have a number of, this is orders. This is the order Caudata, the salamander order. We have a number of different families that radiate out from, that stem from that. So we can represent our different families. And here are our different families. There's one Proteidae, Hyenobiidae, Cryptobranchidae, all these different families we have in here. There's one that's actually called the salamander group there. And from the family, we get different genera. So here are the different genera from a particular family in here. And from the genera, we get individual species that might come from that. So this might be genus Ambystoma, the mole salamander group, and in that Ambystoma, Maculata might be one of them we're picking on there, the spotted salamander, macula, spots. So anyway, we've got this whole series of groups that break out into other different groups, and that's kind of what our naming system kind of makes us see this as. That there are only this many groups within this one kingdom, there are this many groups within that one phylum, there are this many groups within that one order, and so on down the line. But anyway, that's kind of how the naming system would have us classify things. And we know that that probably doesn't represent the way that things would happen in a proposed evolutionary timeline. We would see something more like this, where we'd see different groups break out at different points, a lot of them going extinct and not persisting on into the future. So anyway, all the ones that reach all the way out into here we have our final groups in the end. So whatever we had here, this may have been Ambystum immaculatum. And this one over here might represent the same thing. So anyway, this continuous kind of branching pattern is much different than what's represented by thinking of things in terms of kingdom, phylum, class, and order, family, genus, species. So it represents what the actual history may have been in terms of an line of ancestry. So that's our first trouble with taxonomy. Now on to the second. Okay, moving on to our second trouble with taxonomy. We've got number 2 here, the arbitrary assignment of groups. Groups meaning your different taxonomic names like genus, order, family, so on. So what we're talking about here is if our understanding of the relationships looks something like what we talked about before, where you have a few groups breaking out back here as different taxa, and as you move through time, you see more represented forms. And we have a number of different named species over here. I'll just number them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15 species that are current living forms in this phylogenetic group. So, that's a good number to work with. But look at this chart. How are we going to decide what is going to become, what's going to be our line for the genus level? If we put our genus level, say, right through here, make a line across here, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, almost 15 different genera there. So anyway, the only ones that break out beyond that at the species level are 10 and 11 and 12 in that chart. If we change our genus position back to here, instead of having 14 different genera, we're going to have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So are we going to have 10 genera? Or are we going to have 14 genera? What traits are we going to use? What physical differences, derived conditions are we going to use to separate out our groups over time? So how are we going to draw these lines? And they may draw it in one position for one group of organisms and another position in another group within this same lineage here. So anyway, are there 10 genera or 14? And you go back further, you have families. Back here somewhere, say these are going to be families right here. We'll identify 1, 2, 3, 4, 5, 6, 7, 8, 9 families at this point. But if we go back a little further, back right here, we've got 1, 2, 3, 4, 5, 6 families. So it's completely arbitrary. You'll have scientists who argue their position in favor of one versus another position for separation of taxa, but these names can be put in any place that they decide, whatever they think happens to be best, and there's not always agreement as to what would be best, so you can get different numbers of subgroups depending on who's doing the classification. So arbitrary assignment according to somebody's opinion, where those lines should be drawn to separate out how many genera, how many families, orders, and so forth are in a particular phylogenetic lineage. Now on to number 3. So our third problem or trouble with taxonomy, number 3, hierarchical assignments that are out of sequence. Let me explain what I mean by that. If we start down here with the branching pattern of phylogenetic descent, different groups coming off at different points over time on this family tree of taxa here. So we've got this lineage going on here. Say this over here, these are our phyla here. Class, different classes right here. Say we draw a line for orders right here, so all of these are new orders. Then we have families beyond that. Then we have the genus level somewhere in here, and then we have different species, and then somewhere out of one of these species down here at the end of this chart, we get another group to come out. Let's say it's a new order. A new group comes out here, and a whole new taxonomy of a new order develops out here. But you're saying, but wasn't that just a species a minute ago? And that's kind of the way it is with all of this. You think of this individual right here, it was at some point a species, right? It came along through here. All of these individual lines are represented by individual species, though they also represent these other higher taxa. But you think of how they proposed that birds came out of reptiles. So maybe these are birds and this was a reptile group right here. So we have some kinds of reptiles, some subgroup, family, genus, species, and then from within that a new divergent group comes out, and we're going to call that an order. Even though it may have come from one species or from within a genus or something of that sort, we have a whole new order of organisms coming out there. In the same way we had, let's say, class Amphibia coming out of the Sarcopterygian lineage. It had to come from some particular kind of origin, some kind of species of origin. So you have these things that are not exactly in the right order in the family tree. When you think of them as phylum, class, order, family, genus, and so on. So you kind of have a jump back in taxonomy. You were going to more and more specific things in this phylogenetic chart, and then all of a sudden a new order or a new class or something of that sort crops up from a species within your arrangement there. So things are a little bit out of sequence in that way. Now on to the fourth trouble with taxonomy. Okay, number 4, inconsistent standards across taxa. Inconsistent standards for how we're going to group things. In vertebrate organisms, things that have a skeleton, we usually look to skeletal features of the taxa to help us to identify those important traits for grouping classes, orders, families, genera, and so forth. So we can, if we have skeletal features, if we have hard features, that's the preferred standard to use, because we can refer back in the fossil record and see the fossilized bones of other organisms and make judgments about their placement in taxonomy as well, based on the fossils of skeletons of extinct organisms that are out there. But if you're talking about soft-bodied organisms such as annelid worms, any other kind of soft-bodied organism, cnidarians and so forth, you can't use those same kinds of features. They don't have a skeletal system to use as a basis for your taxonomy. So you're going to be looking at other features. So that the features you use for taxonomy in one taxon of organisms, you can't use that same standard on another group of organisms in the taxonomic scheme. So we kind of are inconsistent from one group to another as to what kinds of anatomical features are going to be vital to the taxonomy of the group. So inconsistent standards across taxes, number 4. Next, number 5. All right, our 5th problem we have with our taxonomic system is only down there at the species level when we're talking about what is a species. Problems with the species concept. The species concept. So I think what we'll do is we'll make another list of some specific things that have to do with problems with the species concept. And that will be our last of our identified problems we have with taxonomy. Misrepresentation of the actual branching pattern if we divide it into just a specific kingdom, phylum, class, order, family, genus, species. Arbitrary assignments of the groups, where do you draw the line, was the second one. Hierarchical assignments out of sequence, so you have orders that come from species and families that come from genera and so forth. And their next, inconsistent standards across the taxa. What you use for the basis for your taxonomy in one group, you can't translate that usually very well over to some other groups. And now problems with the species concept. Let's give those in detail. Okay, problems with the species concept. Most of us have a fairly good idea of what a species is. We know what a robin is when we see a robin. We know what a tiger is when we see a tiger. Most organisms we encounter in nature are easily assigned to a species without any confusion about what kind of organism it is. Think of the normal gray squirrel, for example. Fairly common organism, and they all can interbreed with one another, right? Males can breed with females as long as they're of that same kind from that same species lineage that we're talking about. They produce viable offspring and they have offspring, and on it goes within that group. There's some places where this whole concept kind of gets difficult. We all have heard of hybrids, where one kind of organism crosses with another kind of organism to produce some kind of maybe intermediate looking thing. Sometimes it's difficult to discern the viability of hybrids. While horses and donkeys can interbreed and produce a mule, mules can't generally produce any more offspring. So that's kind of a sterile offspring condition. There are a lot of different kinds of organisms out there that we don't know if they came together, would their offspring be a viable type of offspring? Would that offspring be able to produce more and more offspring out there? So that's our first one there, discerning the viability of hybrids. The second, populations mix in some areas but not others. Populations mix in some areas but not in other areas. So two species come together, say there's a line here where the species A is on this side, species B over here. And in this part of the distribution over here, species A and B will interbreed with one another occasionally and produce fine viable hybrids, so that kind of almost erases the species separation between populations A and B. But down here in this area down here, the two species come together and they encounter one another, but they never interbreed with one another. So there's no crossing in this area. Should they be considered separate species, or should they be considered one species? They're obviously different in some respects because these can't interbreed down here. It's not all that uncommon a condition to see that happen, where they'll interbreed in some portions, other distributions, and not in others. Number 3, what we call ring species. And other examples we call species gradients. I'll begin with talking about our ring species. Say we have here, this is the Himalayas, a big mountain range. There's a species of bird, this is an actual example, a species of bird where there are populations, let's say this is population A, and they expand to a new region to form population B, and here's population C, and as we go around the mountain in the lower elevations, we see expanding populations of this bird. Let's say that A and B can interbreed, B and C will interbreed, and so forth. These can all interchange. They're freely interbreeding populations. They're not different enough genetically that they can't interbreed with one another. But when we get around here and comparing H to A, again, these can't interbreed with one another. They're too genetically different. There's some kind of a reproductive isolating mechanism. They will not reproduce with one another. So this is what we identify as a ring species. So should we consider these two separate species, but how do we then differentiate the others within that? So that's a ring species. A similar sort of thing can occur in a gradient, where instead of it being in a circle like this, where the two populations come together there, we just have a species that occurs over a long distribution with A, B, C, D populations as you go across here, and these mostly interbreed with one another all the way down to the end. But if you took a population from that end and compared it to the species A at this end, again, if they were put together to see if they would produce viable hybrids, they wouldn't interbreed with one another. They are reproductively isolated at the extremes of their distribution. So that's what we call a species gradient, where we have a long gradual change from one form to another. So that complicates our idea of the species, what a species is. That's our third example of a complication problem with the species concept. We thought we had it all nailed down, but then there's things that crop up, like ring species and species gradients. Number 4, there are a lot of different species out there that occur allopatrically. That means that they do not occur in the same geographic location, same physical location. So we'll have one species of lizard in one part of the country, another species that looks very similar to it hundreds of miles away, but we don't know if they were put together. They might look a lot alike. If they were put together, would they produce viable offspring? So we don't do enough checking for the viability of allopatric species. Checking for reproductive isolation in allopatric species. It's number 4. Number 5. Clones and parthenogenic species. Clones and parthenogenic species. These things do not reproduce sexually. They do not have the exchange of genetic information. The offspring are simply carbon copies genetically of the parents. This is more common again than you would think out there in nature, but there are a number of different lizard species that are parthenogenetic. That means that they are basically all female species, and when they reproduce, they produce more copies that have the exact same genetic information as the mother. Similar to the concept of clones, where you take a single cell and you produce more, or you take parts of a parent, like a parent plant, and break it off and start a new plant from that parent part there. So we're having reproduction and increase. We have a population of organisms out there, but they all have the same genetic information. How do we consider whether or not that is a true species if there's no interbreeding that occurs at all? What do we do with that? Number 6, varied ploidy. That means numbers of copies of the genetic information of the chromosomes in the cell. So we know what diploid means, and it has 2 copies. That's what we are. We're diploid organisms. If you're polyploid, that means you have more than 2 chromosomes of each type in there. Tetraploidy is a fairly common condition where you have 4 sets instead of 2 sets of chromosomes in the body. So are those forms that have 4 sets instead of 2 sets, are they actually a different species? The gray tree frog is an example of this, where one form of the gray tree frog out there has 4 sets of chromosomes, the tetraploid condition, and there's another example out there that has the normal diploid condition of chromosomes. Are they different species? Are they going to interbreed with one another? So often the ploidy sets up a new kind of organism just because it gets a different complement of chromosomes. We'll consider it a different species. Sometimes they are not able to reproduce effectively with the diploid form if they're tetraploid. So anyway, sometimes that crops up where basically all the genetic information is the same. It's just more of it in the organism. So that varied ploidy sometimes sets up enough difference so that we essentially have a new isolated, reproductively isolated population of organisms in nature. Okay, along with the same concept we were just talking about, where we had problems with the species concept and all that taxonomy and phylogeny trouble we were talking about, we also have lumpers and splitters. I probably could say, instead of and splitters, lumpers versus splitters, lumpers against splitters. This has to do with, again, how we arbitrarily, more or less, group our different species and taxa in phylogeny. So we have some people who want to put more organisms together within a taxon. Maybe he thinks that there are multiple populations that comprise one species rather than being multiple species, or that there are multiple species in there that make up one genus rather than multiple genera, and so forth. So let's see, a little detail on this, a couple of points to make out here. First of all, lumpers are reluctant to sanction separation of the different species or taxa splitters quite readily separate their taxa into different groups. Also, lumpers look for evidences that separate groups that are permanent separation of the group. Splitters don't need that permanence. All they need is the evidence for effective recent separation. That means that they no longer interbreed, even though maybe they could interbreed if they were in contact with one another. If they're no longer in the same place, they may separate those groups out. So anyway, if you're a lumper, you want to know that if there are two groups that are now separated, they're not interbreeding, is it likely that over the long haul, over thousands and thousands and thousands of years, that if they were to come back together again, they would still be reproductively isolated? If they were truly separate, then they'll allow the separation of the group. Otherwise, they're going to be lumped. So, again, this goes kind of back to the problem with taxonomy, and we have different opinions and ideas about how that should be done, lumpers versus splitters. A couple of examples of this, the what used to be called Audubon's warbler in the west and the myrtle warbler in the east. Very similar birds. One had a yellow throat, one had a white throat. Otherwise, they had mostly the same kinds of markings, but they were considered different species for a long period of time until they found that there were some birds hybridizing up in the northern states. And after they discovered that, then they put them together into the same taxon. It's now called the yellow-rumped warbler. Throughout its distribution, even though we kind of know that there are two different varieties, a western variety and an eastern variety. So they were lumped together, so the bird people that were working on that were more lumpers. Thinking of other examples here, what would be considered a splitter? Okay, now I have an example of splitting, lumpers and splitters. This is one of our local salamanders called a mud salamander. This specific species is called Pseudotriton montanus. Pseudotriton montanus, that's the genus species name. But this population is separate from all those that are in the south and down on the eastern seaboard and along the more eastern states. This is almost exclusively to Kentucky and the very neighboring parts of some of the other states. So it has been proposed that this is a different species than those others, even though most authorities still lump it with Pseudotriton montanus. Some call it Pseudotriton diasticus. So as a subspecies, it would be Pseudotriton montanus diasticus. But anyway, it could be either lumped or split, depending on your opinion on the animal.